Okay. Ladies and gentlemen, good afternoon. Welcome to the Industry Theatre, presented today by Northwest Biotherapeutics. It seems hard to believe that it's 3 years ago since we were here in this very same place. Sometimes when I walk in here, it feels like I've never left and that the outside world is sort of a fiction and that all we do is live here in the ASCO exhibit hall. But then fortunately we step outside and have a nice dinner and get back to reality. So I'll be talking about a very exciting subject here. As you know, the trial results from our phase 3 trial were recently released at a scientific meeting. But today I will talk about another subject that's very exciting to all of us at Northwest Biotherapeutics, and that subject is at the heart of where we go next, the next steps, if you will. We want everybody to be aware also that we have videos playing in our booth, and I think you will find those very interesting as well. So I invite you to come over and visit us there. Now, I want to talk about manufacturing of DCVax-L, which to some of you may be a bit of a boring subject, but manufacturing of autologous product presents very unique challenges that are quite different from what other drug product manufacturing processes encounter. And we need to deal with every single one of those in a step-by-step manner, both as it evolves over time and as our criteria change in their quality requirements as you move from early stage trials to post-trials applications. So I want to talk a little bit about the past of DCVax-L, how it was manufactured in the beginning, how we got started on this whole project, try to take you back in time a little bit, help you sort of relive the past with me. Then we'll talk about the present, where we are now. And then we'll talk about a very exciting aspect of the future of manufacturing. And for that, Shashi Murthy, who is with Flashworks, a wholly owned subsidiary of Northwest Bio, will help me and provide some of the supporting information and some things that we have been able to allude to in some press releases, but have not been able to talk to you about. So I hope you will find that aspect interesting as well. Now, this always happens to me. There we are. This is the obligatory disclaimer. I'm not going to read it to you, and you don't have to read all of it either, but it is important that we put this up for security law purposes. Now, how did this whole thing start? I joined Northwest Bio in 2000, and in 2001, we managed to team up with Professor Dr. Linda Liao at UCLA and her team, including Dr. Robert Prince. And they were working at that time on an exciting application of immunotherapy to brain cancer, which was a completely novel concept because the brain is supposed to be in an immunoprivileged environment. So the immune system can't get to the brain. So why would you apply immunotherapy to brain cancer? The concept that people began to realize at that time is that activated T cells actually can get to the brain. So if you have a mechanism to activate T cells against a brain tumor, those T cells will travel to the brain and they can attack the tumor there. That was a new insight that they arrived at, and then they said, okay, well, how do we activate T cells? T cells you activate with dendritic cells, also new insight at the time, something we were working on under the guidance of Al Boynton, then at Northwest Biotherapeutics for prostate cancer. So working with Dr. Linda Liao was a natural fit for us. Then we would have two organ systems that we could be active in, but using the same modality, the same treatment modality, namely dendritic cells made from the patient, attacking the patient tumor specifically. So it's an autologous product in more ways than one. It uses both the dendritic cells from the patient and it uses the antigens from the patient combined to form the drug product that then activates the T cells if you inject it. But when Dr. Linda Liao was making her product, she was actually using cultured brain tumor cells that she got from surgery, and she put them into culture. And the culturing process could take 2 to 6 months sometimes. And in the life of a brain cancer patient, glioblastoma patient, especially at that time, that is a very long period before you can get to your treatment. You want to be treated now, and you can't wait 3 to 6 months. It was an interesting experimental setup, but not feasible to use for commercialization. So we did some investigation into the product that she was making, and we found out that the acid elute that she was using a low pH buffer to elute small peptides from the surface of the cells, actually was killing the cells. And so what we ended up with was the realization that if we just take killed cells instead of that elaborate process, and we can take those killed cells directly from the tumor, then we have a much more streamlined manufacturing process that we can apply much, much faster, and we don't have to go through the culture period. We changed the product and we changed the process, and then she and her team did another phase 1 trial, phase 1/2 trial. It was really also an efficacy trial, and found that the results were pretty much comparable between the two. And she saw efficacy both in newly diagnosed and in recurrent disease. And that product then formed the basis for the phase 3 trial that we started later, once we had gone through all the negotiations with the regulators and got our clinical sites up and running, et cetera, et cetera, et cetera, everything you need to do a clinical trial. Like we said, those trial results were recently presented at the New York Academy of Sciences conference called Frontiers in Cancer Immunotherapy by Dr. Paul Mulholland. We really appreciated that. And just in summary, I'm not going to talk about those data, but just in summary, we found clinically meaningful and highly statistically significant survival benefit in both newly diagnosed and recurrent disease. And this is with a systemic therapy, and truly, at least in my personal objective, presents a breakthrough in the field, because in 20 to 25 years this has not been accomplished by anybody else. The only trial that's shown some good survival results recently was the Optune trial with the helmet, the device that people have to wear for 18 hours a day. Safety profile is excellent, so the next steps are preparation towards manufacturing scale-up, because that's where this all leads. I still don't know where to point this thing. Okay, so let's talk about the present. What are we doing now? And then we'll, of course, segue into the future kind of automatically. So this is our process currently. I don't think this pointer is, yeah, it does show up actually. And I'm sorry, this slide did not quite come through very well, so it's really hard to read. But it starts with removal of the tumor, surgically removal of the tumor, and then we ship that tumor to the manufacturing facility. We ask the surgeon to remove some of the necrotic material while in theater, and then put it into a digestion buffer that we've provided to the site. That digestion buffer then digests the tumor into a single-cell suspension. That single-cell suspension is then centrifuged and subjected to freeze-thaw. And freezing and thawing and freezing and thawing and freezing and thawing cells lyses the cells. It bursts the membrane, so the proteins just flow out of the cell. And you end up with a protein suspension. Those proteins form the antigens that you then use to load the dendritic cells. Now, how do we get the dendritic cells? The patient actually then, after recovery from surgery, undergoes a blood draw, a leukapheresis. It's about a 3 to 4 hour process where we run the blood through a machine and we collect the white cells out of the blood. And the white cells include monocytes. And monocytes have the wonderful characteristics that they will stick to plastic. And so they will do that exclusively. Other cells in the blood don't do that. So that's a very simple and effective and very clean purification process, because you can stick the cells onto plastic, wait a couple of hours, wash off the other cells, and what you're left with are pure monocytes. Then if you feed those monocytes with 2 cytokines, namely GM-CSF, granulocyte macrophage colony stimulating factor, and interleukin-4, those dendritic cells then differentiate, those monocytes then differentiate one by one. They don't proliferate, we don't get more cells in the process, they just become their more mature counterpart, namely the dendritic cell. Dendritic cell, when we at that stage harvest it, is still what is called immature. Immature dendritic cells are really good in taking up antigens. So those immature dendritic cells are then fed with the proteins that are coming out of the lysate. And that, if you let that sit overnight, those dendritic cells take up the proteins and start expressing them on their cell surface in the context of what we call MHC, matrix histocompatibility antigens, which primes the dendritic cells for interaction with the T cells. Those dendritic cells we then freeze, and we have at that point typically between 10 to 20 treatments for that patient in the freezer, and they last for a very long time, and we have long-term stability data on those products. You can call those educated dendritic cells because they now know what it is that they should direct the immune system to. What the immune system should attack is now known to those cells. And those, like I said, we can store, and we can ship them to the clinical site, and there they can be thawed out and directly injected into the patient without any further manipulation. So we don't put any burden onto the hospital or onto the pharmacy other than storing it, taking it out, thawing it, drawing it up into a syringe, and injecting it into the upper arm. I always think that's kind of funny. We put it into the upper arm because we think the arm is closer to the brain, so therefore it might work better, but you could probably put it into your leg or your big toe, and it might just as well, because the dendritic cells have to go to a lymph node, and it doesn't matter where that lymph node is. So there's a couple of characteristics here. It's a very robust manufacturing process. It actually works really well. And we've also found that because we had to activate a manufacturing site in Germany for the clinical trial in Germany, so we transferred the whole process. And it turned out that with one cycle of technology transfer of the documents and training, they could manufacture the same product with the same characteristics without any problem. So it's also robust from that perspective. It's a quick process. It's an 8-day process. It's a relatively low cost as far as cell therapy products go. And the yield is that we have about 3 to 5 years of doses in a single batch. And so it becomes for that patient, in essence, an off-the-shelf product. Now, I love this picture. Mike Scott gave this to me earlier. I think the person who's actually doing this is A, completely unrecognizable, but B, probably in the audience here. And you can see that how this is being done. It is at this stage still a manual process, and you can see the dedication in the eyes of the person who's doing this. It takes trained technicians, and those people that are trained at this are really good and are extremely dedicated to the quality of the product. They know that what they have in their hands could be potentially be life extending for the patients that it is we're going to be treating. So they're it's an intensely dedicated team, and I'm really proud to know at least some of them. I know it's a lovely picture, but I don't have to stare at it all day. So what are the typical industry challenges for an autologous cell therapy product? Because this is different from making antibodies. This is different from making any drug, because we have to make the product for each individual patient. The product doesn't work if it comes from somebody else. The dendritic cells have to match the T cells, and the antigens have to match the antigen in the tumor. So you have to make this product for each individual patient, which means that each individual patient needs to undergo a round of testing before you can even allow the product to be obtained and further manufactured. Testing for virus infections is a very obvious one, so that's one thing we have to do. Then once you start the process, you want to be able to demonstrate that you're controlling the process, and that you're controlling the process every step of the way, and therefore you need in-process controls. You want to know that the cells that you're adhering to plastic are actual monocytes. You want to know that the cells that you're harvesting on day 7 are actually immature dendritic cells. You want to know that the product remains sterile over time during the process, et cetera, et cetera, et cetera. And you don't know all of that at the time that you start your manufacturing process. There's a lot of learning going on while you're doing this, and therefore you have a series of assays that tend to improve during the course of clinical development. And you can use those assays to demonstrate consistency of the process, not only during the week that you're making the product, but also over the time that you have developed your process and developed your clinical trials. And this is extremely important, and it is something that regulators then are also require you to do. So it's something that we have spent a lot of time and energy on, and we believe that we have really good control of the process, and that we can actually formally demonstrate that using these in-process controls. Now, that is only half of it, because now you've defined your process, you've controlled your process, but what about the final product? So there basically the same thing applies. You need a set of assays that again tend to evolve over time and tend to improve over time, that demonstrate a few aspects of your product that are really required in order for you to be able to release it into administration to a patient. One thing that you need, and this tends to be a big bugaboo for people who work on cell therapies, and we believe that we have an adequate solution for it, is a potency assay. The potency assay needs to demonstrate that the cells are actually capable of doing what you claim that they're doing. And in our case, we claim that our dendritic cells are capable of activating T cells, such that those T cells are then capable of killing tumor cells once they arrive in the tumor environment. Setting up an assay to demonstrate that is by far is absolutely not a simple thing to do. And we started with this back in probably 2002. We had a wonderful guy named Gopi Shankar in our quality assurance department who developed the first potency assay for DCVax, and we're now seeing some evolution of that assay and some further improvement that adds some more specificity to the assay. And I'll get to that in a little bit later. Now, those assays also help you to establish long-term stability, because when you show long-term stability, you have to show that every time that you thaw out a vial, after 1 year, after 2 years, after 3 years, whenever, that it meets the same criteria that it met initially when you froze it down. We find that these cells are extraordinarily stable at the liquid nitrogen or vapor phase nitrogen temperature where we store it. And we have satisfactorily established long-term stability. But that's another aspect that people tend to sort of forget about until it's late in the game, and then becomes difficult to establish long-term stability if you're just starting to develop your assays. And then, of course, you need proper packaging that is suitable for end-user use. There's an R too many in there. And it has to be scalable as well. And we have... We're now working with a new automated filling system that we believe meets all those criteria. And then, of course, when these products are shipped, it's not a box with 1,000 vials that you're shipping to a pharmacy that they can store somewhere at ambient temperature. All of these products need to be shipped at ultra-low temperature in vapor phase nitrogen so that the cells remain viable, because don't forget, we're shipping live cells, and those cells still need to be alive when you inject them into the patient. So the cold chain maintenance for the whole process is critical and essential for the quality of the product. This is a picture of the automated filling system with one of those vials here. These are eminently suitable for use in pharmacies and at the bedside. They're glass vials. They thaw out very quickly. We have a small amount of product in there. They have a punchable rubber seal, so all you need to do for the nurse is the common process of just drawing up a little bit and injecting it into the upper arm. Now, let's talk about the potency assay a little bit more. Dendritic cells are somewhat unique. They're antigen-presenting cells. They're not the only antigen-presenting cells. Monocytes can present antigen as well. B cells can present antigen. But dendritic cells are unique in that they're the only cells that can induce what we call a primary immune response. They can induce an immune response where none exists at that point in time. So if we want to activate somebody to mount an immune response against their brain tumor, that's exactly what we need, because that immune response does not yet exist. Brain tumors are different in that sense from tumors that arise in the skin or in the kidney that are highly immunogenic. There you have a natural immune response that evolves with the tumor over time, and the tumor learns to protect itself from that with checkpoints. You can use checkpoint inhibitors to treat those diseases. With brain cancer, you can't do that because there is no pre-existing immune response. We need to induce it, so therefore we need these dendritic cells because they are the only ones that can induce a primary immune response. And they do that by delivering an activation signal to T cells. It's one of two signals that is being transferred. In essence, there's actually three signals. The third one is a soluble cytokine. But this is what is called the co-stimulatory signal. So the T cells need actually three signals. One is the specific triggering through the T cell receptor. That is the receptor by which the T cell determines its specificity. The other one is the co-stimulatory signal that comes from the dendritic cells, and then there's the cytokines that tell the T cells which direction they should differentiate in. So we don't have a T cell trigger, T cell receptor trigger in this assay, so not a specific one, but we use an aspecific one that works just as well. A little bit of antibody that you add to the assay that triggers the T cell receptor. So that's signal 1. Signal 2 is the co-stimulatory signal. Once you have those two signals, the T cells start to divide, and you can divide the number of divisions, you can measure the number of division cycles that these T cells go through. So that makes for a beautiful assay. That was, like I said, developed by Gopi Shankar. And then Shashi and his team made some further refinements and improvements to this, which actually adds to the functionality of this particular assay. And we're very happy with that result. There must be a place to point this thing. Ah, there it is. So one thing that Shashi found is that if you do this particular potency assay, you see here the number of cell divisions on the x-axis, and you see here that if you add tumor lysate, that you get an increase in the signal. So now we have a way to measure not only whether the dendritic cells are functional, but also whether they have added functionality after the addition of the lysate. So this adds another dimension to what we already have. Until now, we could measure whether we had dendritic cells that are capable and functional in stimulating T cells, and now we can also measure whether we have actually added the lysate that makes the T cell specific, and that actually results in further activation of T cells, as you can see here. So let's move on. So then the team under Mike Scott and Mark Lodell said, okay, and I'm not going to ask you to read this slide, and I'm not going to read it to you, I'm just going to point a few things out. But because now the question is, can we activate those T cells so that they actually kill tumor cells, and can we demonstrate that? That is a proof of concept question. You can't do that for every single patient. We can apply the potency assay for every single patient. Here the question is a more fundamental one, is if we do this, if we go through this process, are those T cells that we generate actually capable of killing tumor cells? Because that's what we're after. And that is a very time-consuming process, because as you can imagine, if you put 1 million T cells into a test tube, then there might be 1 or 2 T cells specific for the tumor cell that you want to attack. So in order to get something measurable, you need to turn those 2 T cells into 2,000 T cells, which takes a number of rounds of re-stimulation. Every round of re-stimulation takes 14 days. So you can do this for a long time, and then you end up with a population of T cells. And here I show you, and these are the black bars here in every single case, that if you do that, you actually get killing of your target cells, and all the other bars are positive and negative controls, which is why you don't have to run through this. So what we have now is a number of things that come together. I've already shown you this slide. And that is that we have a potency assay that shows that we have dendritic cells that are loaded with tumor lysate, which is exactly what we need for every patient. And we've demonstrated that the dendritic cells that we make are capable of activating T cells to kill tumor cells. So that brings that whole story full circle. Then we need to test whether we have the right cells and whether we have enough of the right cells and not too much of cells that happen to come along in the culturing process. You start with a lot of white cells that contain a lot of T cells and B cells, natural killer cells, and we need to get rid of those during the culture process, not because they're bad, they're not bad actors, but we just want to deliver a product that's as pure as we can. That's in part because of regulatory constraints, but it also is in part because then you can give fewer cells, and therefore you can inject the cells where they belong, namely into the skin. If you have a product with lower purity, and this has happened in the past with other products that were developed based on a similar basis, if you have a purity of 10 to 15%, you need to infuse a very large volume into the body in order to activate the immune system. And the only way to do that is to put them into the bloodstream, but the bloodstream is not where these cells belong. It's not how they get to a lymph node. So doing this in the small volume is critical, and for that the high purity is critical. And we're quite proud of the high purity that we have. And we demonstrate that we have that by using 2 antibodies, CD86 and MHC class II, and we use another couple that I won't go into right now. in order to interrogate every product to see what the purity of our product is. And these two molecules are actually very important because these molecules are involved again in the potency assay. So we try to tie everything together into a logical sort of puzzle where the pieces fit together. If you add these antibodies to the co-stimulatory assay, to the potency assay, it blocks proliferation of the T cells. So these molecules were actually chosen for a very specific reason. And then we test purity by testing for B cells and T cells and NK cells. And again, we've shown that this is all quite reproducible. I have a few slides to show you. This gets a little technical for the people that are not used in looking at these, but there's two antibodies here that are used to stain the cells, the dendritic cells, and everything in this quadrant is positive for both of those markers. So you see that if we make dendritic cells, that almost every single one of them falls into this box. I think the purity of this one, Shashi, was like 98% or so. Yeah. So that was a very nice, very good product. But if you use the T cells from that same product, a little bit of T cells is still there, you can see that those do not stain with those particular antibodies. So those antibodies are really specific for the cell type that we want. And then this is just a typical profile where with a series of antibodies we demonstrate that the cells stain positive for these particular antibodies that we're using in that same assay. And this is a routine picture that we use for every product that comes by, and this all goes into the computer and the paper record and is stored with every product. So we can always go back to it and verify that we're doing the right thing. Now, there's a manufacturing facility in Sawston. It has over 90,000 square feet, over 2 floors, and only 20% of this is built out. I'll talk about the capacity of this facility next. It is a good global manufacturing hub. It's close to airports and is very well suited for purpose. That one. Okay. Here you can see the first floor facility, and in purple, that thing that just flew in because I have poor control over this thing here, is the part that's actually already built out. And this is only 1 floor. So about 20% of the total facility is built out. And when we get to the capacity question, I can answer that as well. Currently in that facility, we can manufacture up to 50 treatments a month in 2 clean rooms. And this can be scaled up to the actually quite large number for glioblastoma of 1,000 patients per month. And the storage space is really impressive, of up to 3 million vials. So for now, I think there's sufficient storage space in that facility. Other finger, maybe? Yeah, there we go. So this is just a picture of the storage space. These are those big upright freezers. I don't know if you remember, but in the beginning of the COVID pandemic, there was suddenly an urgent shortage of these freezers because everybody needed to store the COVID vaccines, and they were nowhere to be found. But quite a few of them are here. So the team that we have has a lot of expertise, both in process development and assay development. We talked about that a little bit. Using in-process quality controls, establishing product release assay, product potency assays, are always working in very close contact with the regulators to make sure that whatever we do is in very close alignment with what regulators require. Digitization of the manufacturing process chain. What we're used to during the clinical trial is that for every patient there is a booklet of paper that has to be filled out manually. It's doable, but it's not very user-friendly. And we want those dedicated technicians to spend as much time dedicated to the product and as little time doing administration. So digitization of the manufacturing process with barcodes and things like that is critical to the success of this project. Gold supply chain management, from when you take things out of the patient to putting it back into the patient, from vein to vein, as they say, is critical, and the team has great experience in that. I mentioned already extensive regulatory knowledge and expertise, and they have also experience in statutory licensing by the medicine and tissues regulatory agencies. A great team to work with. So that brings me to the next topic, and that is the future. And the future, we think, is in process automation. And And process automation in cell therapy is a particular challenge, and it's again a different challenge from having just basically setting up a filling station, because, and Shashi will go into the specifics of that much more. But let me just, and it's also a topic that's very near and dear to my heart. I'll talk a little bit about some of the automation processes that we developed before Shashi came on board, and then he will talk about the stuff that his team has done. But I think it's really it's a critical endeavor that we're embarking on, and we're well underway in making this successful. And just to like paint you a picture, if you saw that person in the clean room pipetting the solution in and out of that tissue culture flask, just think about it. When we make a DCVax product, it takes X number of hours for 2 people in the clean room with somebody from quality control standing outside monitoring the process, and that's over 8 days, and that's for one product. Now, it's manageable, it's doable, it turned out to be very effective. When you look at the clinical trial results, we can't really complain about it too much, but if you want to scale this up, you want to bring down the cost of the product, then you want to automate. And think about what that would mean, because now we have 2 people, let's say, rough number, that spend a lot of hours in the clean room for one product. Instead, once you have this automated, you can take this into a less controlled environment, not completely out of the clean room environment, less controlled environment, and one person can supervise a number of machines that are making products in parallel. Imagine the cost reduction and the scale up that you can accomplish by doing that. So we worked a bit on a different product called DCVax Direct. I had the honor to talk to you about this in the past. But this was really our first endeavor into automation of manufacturing, and it was set up a little bit differently because there was no existing process that we needed to mimic. We could start from scratch and say, okay, if we want to make a product in an automated system, how do we want the system to look so that the product we get out at the end is biologically optimal? So we weren't constrained by expectations. It has to do this first and then do that first and then that first. We had the free hand, which was very liberating. And so we developed this machine called the TFF, Tangential Flow Filtration Cell Separation System. And it's sort of an anti-gravity machine, if you will. There's a fluid stream under a filter, and then there's a fluid stream gently through the filter, and only the small cells go through, and the large cells get left behind, and the large cells contain the monocytes. So then you can just put that into a bag, add your cytokines, and then activate the cells, and that's your final product. So a very simple and I think beautiful process. But like I said, that was set up without any constraints. It just, the only question was, can we get a good product out at the end? The situation that Shashi found himself in was very different, and that is that there is an existing process, and now how do we automate it? And how do we automate that such that we can actually make that a viable process to apply to a very large-scale environment? Because remember, we're talking right now about applying DCVax-L to brain cancer, but it's not a product only for brain cancer necessarily. In principle, any solid tumor can be treated with this. We've done a small trial in ovarian cancer, and we've done quite a few patients on compassionate use basis with different tumors, and some of those are actually sitting here and joining us in this hall. So we have to think big, and we need this process automation to allow us to think big. And so with that, I'd like to hand the microphone to Shashi, and then he'll talk about the Flaskworks project.
Great, thank you, Marnix, and hello, everybody. I'm going to try to figure out how to use the pointer first. This is good. Nope.
It is that button, yeah, that button.
Great.
Yeah.
So, how many engineers does it take to turn a light bulb on? So, it's that kind of thing. So, okay. In this case, more than one. So the goal of our work within NWBio is to replace a manual process with an automated one, as Marnix has suggested. The process, as it stands now, begins with a centrifugation process to isolate peripheral blood mononuclear cells from a leukapheresis product. And this is done using something called Ficol separation, which is a well-established process. But what what is relevant to this discussion is the fact that it's performed in containers that are opened. Liquid is moved into those containers. The sample is then centrifuged, and then you draw a suspension of cells that is then plated into culture vessels of different kinds, that are then manipulated on an almost daily basis manually. And at various points of the process, the vessels are opened, for other manipulations, and that's how the process is carried out over 8 days. Now, as you can imagine, this is fairly labor intensive just in the mechanics of the process alone. And then furthermore, when you add the requirement that this has to be done within a clean room by a very skilled operator, you layer on all kinds of risk as well as cost. And so, what we're aiming to do here is cover the entire spectrum of the process, starting from the very beginning, the isolation of the peripheral blood mononuclear cells, and then the subsequent culture processes. Now, the good news is with respect to the initial part of the process, namely the centrifugation portion that I mentioned, there are a number of commercial off-the-shelf tools available to do this, and so we don't need to reinvent another process to do that. And so we focused our attention on the culture portion of the process. So what does it take to automate? This is actually a fairly involved process in any industry. If you think about the diversity of products you can get at a Starbucks, and then you look at the number of machines they have, and then you look at the number of people they have, it's actually quite a few. So it's not like there's one magic machine that gives you every product that you could possibly need. And so in the context of cell therapy, automation of cell therapy manufacturing is a huge challenge industry-wide. In the realm of personalized or autologous cell therapies, the challenge is further compounded by the fact that the starting material is different every time. It's not exactly the same specific kind of material that you might expect in, let's say, monoclonal antibody manufacturing. Furthermore, cell therapies are of all different kinds. You have CAR T, you have NK, you have allogeneic, you have autologous, and you have dendritic cell-based therapies. And the culture of cells, by their very nature, tend to be dependent on what it is you want to do with those cells. And so every therapy's process is unique and therefore requires a unique automation approach. And so with all of that in mind, we brought that kind of thinking to the manufacturing of DCVax-L. And really what that involved was looking at all the different steps in the process and examining how we could automate them and then ultimately put together all of the automation elements. So, I've shown sort of a summary of the current process in the top here. You plate the cells in a vessel, wash away non-adhered non-monocyte cells, differentiate the monocytes into immature dendritic cells using IL-4 and GM-CSF, classical process, harvest those cells, and then treat those cells with tumor lysate, plate them again, harvest those cells, and then you cryopreserve them. This is already quite a few steps here. When you add on to the fact that when you're carrying this process, you're manipulating or you're opening and closing vessels, there are what are called open and manual steps. And those are shown in the red arrows on top. The big takeaway here is that there are many red arrows in the first row. That's all. If you remember one thing from this presentation, the guy said there was many red arrows. And we want to get rid of those red arrows. You're opening and closing them. There's risk involved. It's very manual. You think about the operator in a full suit, in a clean room, working at a biosafety cabinet. This is very, very, very involved. We're not even thinking about cost here. Cost, infrastructure, all of those things are very complex. So when we think about an automation process, we really have to think about every one of those steps. Adding the cells, washing away the undesired cells, culturing the adhered cells to where they need to go, removing those cells, treating them with tumor lysate, removing those cells, and then bringing them on to final product fill and finish. It sounds very prosaic when I say it like that. You bring cells in, you culture them, and I didn't even talk about washing them. That's a very important thing you have to do. Loading with lysate. Every one of these things has required innovations in technology, and this is what we've been actively working on over several years now. There are 13 patent families associated with let's see, 4 bullets here. So we've successfully accomplished those steps, which is we think is a big milestone. But furthermore, that's not it. Like once you actually get an automated process going, you're not done. It's far from done. You have then designed a customized instrument. You have designed single-use disposables, and you can't just have one disposable that somebody glues together in the back room. You can't do that. You have to have systematically manufactured disposables that have a security of supply, whose parts are actually regulated for medical use, and that can be properly sterilized to avoid contamination risk, and are actually available when you need them. And so there's a picture of a disposable at the bottom there, and I think you can see that contains several different kinds of tubing. So there are, in fact, several different kinds of tubing there, each of which has a specific role, function, characteristics, length, width, and material, all different kinds. And just to kind of give you an idea of the complexity of this, that disposable is made for us by Saint-Gobain. If you think, Volvos and BMWs, the windshields, this is the company that did the glass at Versailles, 400-year-old company that does very, very good work across the globe. So that disposable is made at 3 different sites at Saint-Gobain. It's a little bit like the Detroit auto manufacturing, where you have one part made somewhere, it's brought to another place that something else is done to it, and then the assembly is taken somewhere else. And that's just the disposable here. And so that is the kind of level of detail that one has to bring to an automation process to begin with. And again, these are just the bare bones mechanics of the whole thing. The next steps in the road ahead, there's a lot more to do here in terms of finishing out the automation of the process. But then ultimately, we'll have to move on to implementing this at the manufacturing setting and taking this through the various validation and comparability testing steps that we need to undergo. So with that as the sort of the path ahead, I think I want to sort of summarize by highlighting the benefits of automation. Really, it's worth reiterating this in light of where it is we're headed. The goal is to reduce operator time and be able to run more processes, create a consistent product so that every time you process a leukapheresis product, you get the same thing. And that the machine can provide the same thing, and it can be handled by an operator who doesn't need to have extensive training or experience in handling cells. And furthermore, we want to be able to realize the ability to reduce the stringency of clean room specifications. Clean rooms are very expensive to build, very expensive to operate, and very expensive to have people working in them. Not to mention the level of just the sort of the sheer physical exertion of working in such spaces. And so really that's where we're headed in terms of being able to design instruments that can be run without supervision, that can be started up and then left to run for a portion of time, 7 days, until the next step, the final step of the process needs to be carried out. And this then allows operators to start multiple machines at the same time, leave them, and start different machines at different points in time for staggered manufacturing. And be able to run these without the need for constant presence in the clean room. And so these, I think collectively, is what will enable us to realize significant cost reduction as well as very high level of scale up. So with that, I'll hand it back to Marnix to wrap up.
That was great, Shashi. Thank you so much. Really appreciate it. So to give you just a quick summary, the phase 3 data, like we already said, were presented in May. And we have now been working very hard, and I hope that I gave you a little bit of insight into this, into establishing manufacturing capacity to support scaled-up delivery. We added digitization of the manufacturing process, and we've obtained and retained in-house expertise in process development and product testing. And what that entails, or in summary, if you want to have a good catchphrase to take home with you, is that we've solved the 3 Ps: procurement, production, and the patient supply chain for DCVax-L. And I can tell you that I'm at least, and I think we all are very proud of the team that has accomplished that. So we have solutions to the common challenges of personalized cell medicines, and we just gave you some insight into some aspects of that. And with that, of course, when we talk about past, present, and future, the future actually is already here. So with that, I want to thank a few people. The clinical trial, of course, couldn't have been run without the patients who were willing to participate and subject themselves to the treatment that we thought would be good for them, and the families that have supported them over time. All the investigators in the clinical trials, we had a lot of sites throughout 4 continents, and we're extremely grateful for their support. When you come in there as a small company that has a complex problem to present, namely autologous cell therapy for brain cancer, you need all the cooperation you can get from the investigators, from the nurses, from the regulatory specialists, from the IRBs and what have you. And I can tell you that that support has been very strong and unwavering, and we're truly grateful for that. Dr. Linda Liao and Dr. Robert Prince at King's College, Professor Ashkan, he was the European principal investigator for the trial, have really sort of held her hand over the past 15 years. Mike Scott and team in Sawston, Shashi Murthy, of course, the people at Cognate when we worked with them, Paul Mulholland for doing the presentation at the New York Academy of Sciences, of course, and of course the team at Northwest Biotherapeutics. So with that, I'd like to round this up, and if there's questions, you can see us in the booth and we'll talk to you there. Thank you really very much for your attention.